Hi everyone, we are from the NSF Engineering Research Center for Cell Manufacturing Technologies, or CMaT. And today you all are going to be learning about a day in the life of two PhD students in biomedical engineering here at UW-Madison who work in cell manufacturing. Some brief introductions. I'm Madeline and I will be joined by Kirthana. We are both second year biomedical engineering PhD students and we work on creating cell therapies. Hi everyone, I'm Madeline. I'm a second year PhD student in biomedical engineering here at UW-Madison. I am co-advised by Melissa Kinney and Chris Saha, and I work on trying to make therapeutic T cells in the lab. Today I'm going to be taking you around the Kinney lab in the BME building here at UW and showing you an experiment about embryonic stem cells. So the first thing that I like to do when I enter lab is to grab all of my personal protective equipment. So that's my gloves, my safety goggles, and then also lab coats. And then we will walk around the lab. So we can see we have a couple of centrifuges and a microscope here. And then more centrifuges and another microscope. And then here are our incubators. So this is where we grow our cells. And then we also have all of our biosafety cabinets. And what these do is this is where we work with our cells to make sure that no contamination can get into them and that our experiment stays sterile. So the experiment that I'm going to be showing you all today when we go back into lab is an embryonic stem cell passage. And basically what this means is that because my cells have been growing for almost a week now, there are too many cells for the dish that they're currently in. So what I have to do is remove the cells from the dish and then replate a much smaller number of those cells onto a new culture dish so that I can continue growing them. So this is what my cells look like before I start to passage them. You can see that they look really crowded. Here's my setup in the biosafety cabinet. Let's take a closer look. So here you can see all of the reagents that I'm going to be using for my experiment today. First, on the left, we have phosphate buffered saline, which is also called PBS, and that's so that we can wash our cells and remove any protein. Next, we have what's called releaser, and that is a reagent that will lift the cells off of the plate so that they're not sticky to the bottom of the plate and I can manipulate them. Then on the right, I have a tube of cell culture media, which is what the cells grow in, and that's kind of their food. And then finally, we have my actual plate of cells that I'm going to be working with today. Now I need to wash my cells to get rid of any extra protein. Then I add the dissociation reagent so that the cells start to lift off of the plate. That incubates for 5 minutes. While that is incubating, I have to prep my new dish to receive those cells. Now my cells have detached and it's time to collect them. So I wash the old plate and I mix it up and add 1/20th to my new dish so that the cells have more space to continue growing. This is my old plate next to my new plate. And don't forget to record everything in your lab notebook. This is what my cells look like 24 hours later. See how they're much more spread out. We also culture other different types of cells in this lab. So I had a really good time naming this incubator Gus from Cinderella. Let's take a look at what my cells look like now. Looking good. Now my lab mate is going to take you around the Wisconsin Institute for Discovery at the Saha lab.
Hi everyone, I'm Kirthana. I'm a second year PhD student in Chris Saha's lab at the University of Wisconsin-Madison. And today I'm going to be giving you a tour of our lab and showing you how we isolate natural killer cells from whole blood. So let's get into the lab and get started. And so now let's get into the lab. So this is one of our cell culture rooms that we share with a couple other labs. This is Saha lab only cell culture room, and I'll show you what it looks like on the inside in a little bit. Here we have our rack of lab coats that we wear for PPE. And there's our microscope room that we use to take really cool images. We have a couple microscopes in there. And this is our main lab space. And so this lab space is shared by 4 other labs, and each lab has their own section. And so down here is the Saha lab. So here we see our bacterial bench, which is a common shared bench, and then these two are grad student benches. And then here's another shared bench with a common piece of equipment. And then here's where we do all of our gel work. And above each bench, you can kind of see general lab reagents and equipment that we need in everyday experiments. And this room is an extension of our main lab space. And so there's more benches, and this is my bench. And I'm in the middle of something, so which is why you kind of see stuff laid out. And one cool, unique thing that our lab does is we put stickers on our pipettes to show who they belong to. And I just think that's a unique touch. And so I have the Strange Planet comics on mine, while another lab mate has different types of dogs to show that they belong to her. And here we have our cell culture room. So our cell culture room is basically where we do all of our sterile work and our cell experiments. And we work in biosafety cabinets, or BSCs, which is what this is. And we have 2 of these in this room. And what these do is basically keep the air inside that glass hood sterile, so any bacteria that could be floating around in the air doesn't get into our samples and contaminate them. So once we're done working with our cells in the hood, we then put them into incubators. And so the top one is reserved for stem cells, and the bottom one is reserved for non-stem cells. And these are set to very specific temperature and CO2 and oxygen levels to keep the cells happy and healthy. So if we take a look inside, we can see some cells are being grown on plates by my lab mate. And we also have a centrifuge. And so what a centrifuge does is spin things down really fast. And so anything solid in your sample will pellet to the bottom, and you can separate the liquids from the solids depending on what you need. And lastly, we also have a microscope that we use to see how our cells are growing on whatever plate that we put them on. And so I'll put a picture of what the cells look like at the scope in the next slide. So before we start our isolation today, I'm going to tell you a little bit about natural killer, or NK cells. NK cells are a part of your immune system, and they act as kind of like your first line of defense in your body against viruses, bacterial infections, and even tumors. And so what they do is they go around your body looking for any signs of a foreign material or a foreign pathogen. And when they find that, they then kill that unhealthy cell. And this keeps you from getting sick. And the way that this works is that the NK cells have an on-off brake system. So when they're going around your body and they come across your healthy cells that are working well, this brake system is kept on. And so your NK cells don't kill your healthy cells. But when they come across cells that are starting to form a tumor or have been infected with a virus, this brake system is turned off. And that lets the NK cells know that this target cell is not a good cell, we need to kill it. And these cells have gained a lot of interest recently in the immunotherapy field. So an immunotherapy is when you use the patient's own immune system to treat diseases like cancer. So a large part of my project is to figure out how can we engineer patient NK cells to do their job better? So how can we get them to detect the cancer and to kill it entirely? And so by using the patient's own cells, we can avoid them having to go through chemotherapy and radiation therapy.
Once we have our blood sample, we add a special cocktail to the blood that binds all of the non-NK cells to the red blood cells, which leaves the NK cells free to be isolated. We also have to prepare special tubes that will help us isolate the NK cells. And we have to add our blood directly to those tubes before we centrifuge. This is what the tube looks like before it goes into the centrifuge. So this is what the tube looks like when we pull it out of the centrifuge. And what the centrifuge did was spun this tube really, really fast so that all of the red blood cells that were bound to all of the non-NK cells were pulled all the way to the bottom of the tube, and this clear liquid on the top has our NK cells, which is what we want. After washing the cells a couple of times, we can then put them in their plate with their media or cell food so that we can start growing them. Thank you all so much for joining us, and make sure to follow us on social media if you want to learn more about cell manufacturing.